Hello everyone, my name is Oliver and today I will be telling you a little bit about C4 plants. So let's get to it. Starting off, we've kind of talked about stoma before, but just as a review, basically they can close at will, or the plant can close them, and they let things like water and oxygen out and CO2 in. This is going to be very important, so just remember this. Now, let's talk about the difference between C3 and C4 plants. So, let's just remember that C3 plants live in much cooler, wetter environments where there's lots of water. C4 plants tend to live in climates that have a lot less water, so they're dry and pretty hot. Now, what does this mean? Well, the C3 plants, where in a very moist environment, can keep their stoma open a lot more because the loss of water out of the stoma is not as big of a deal because, again, they could just get more water. It's not a big deal. Whereas the C4 plants in a much hotter environment have to keep their stoma closed so that they don't lose water because they don't want to dehydrate. Now, what this means is that inside of the C3 plants, the oxygen is going to be able to get out and the CO2 in. And this is good because obviously photosynthesis needs the CO2, and we want the CO2 inside of the leaf. But in a C4 plant, the oxygen can't get out because the stoma are closed, and the CO2 can't really get in. And what this means is that inside of the C4 plants, there's going to be much higher buildup of O2. Now, this is really bad because if O2 gets into the Calvin cycle, we get photorespiration, which is obviously bad because it results in energy loss. So ideally, we want to have as much CO2 inside of the plant and as little O2. But for C4 plants, this really isn't an option because they have to keep their stoma closed to conserve water. So essentially, the biggest difference is that they found a way around this problem. So just a quick, I guess, lesson about the structure of a C4 plant. So this is a C4 plant. What's really important here is to notice these red parts here, the mesophyll cells, and these blue parts here, the bundle sheath cells. So basically, these purple parts are the stoma. The O2 is going to be leaving through here, but of course we know that in a C4 plant, it's not going to be able to leave as much. So we're going to have a much higher buildup of O2. But what's really important about the mesophyll cells is they actually protect these inner bundle sheath cells from getting any oxygen. So just remember this when we talk about this next part. So let's look at this a little bit more closely. This red box here is our mesophyll cell, and the blue box is the bundle sheath cell. Right next to it is our C3 photosynthesis, just for comparison. So in a C3 photosynthesis process, we have RUBP and Rubisco up here in the mesophyll cell. This is just normal photosynthesis that we've seen, so I'm not going to talk about it. But what I do want to point out is that this one carbon here, the CO2, and this 5 carbon RUBP are going to combine to make 2 3-carbon structures. And this is why C3 plants are called C3, because it's this 3-carbon structure. In a C4 plant, we actually have no Rubisco and no RUBP up here in the mesophyll cell because they're all down here in the bundle sheath cell, but more on that in a second. So these C4 plants here, the CO2 comes in and combines with this different thing called PEP, which has 3 carbons. Now this PEP is very selective. It will almost only combine with the CO2. So even if we have a ton of O2, which of course we kind of know is going to happen inside of the C4 plants, because again, the stomata are going to be closed, the PEP will really only combine with the CO2. And this will create oxaloacetate, which is 4 carbons. Now you might remember from earlier our C3, this is why it's called C4, because this is a 4-carbon structure. Now this 4-carbon structure needs to go one step farther pretty much, and it's to turn into malate. To do that, we need the NADPH, which is created from our light reactions. So this is why we have a little sun here. Anyway, so this malate here, after it's created, again, 4 carbons, C4, will diffuse through the membranes into the bundle sheath cell. Again, these are the cells that are protected from the O2. So now this malate is in this bundle sheath cell. It will split to turn into CO2 and pyruvate. CO2 is the exact same thing that came in, except in here we're going to have very, very, very little oxygen. This pyruvate we're going to talk about in a second, so don't forget it. Now this CO2 goes into the Calvin cycle. Here's our RUBP, Rubisco, all that fun stuff. But we know how this works, so I'm not really going to be talking about it too much. What I want to focus on is mostly the fact that this RUBP and Rubisco resides inside of the bundle sheath cell, whereas in the C3 plants, it is in the mesophyll cell. Now this pyruvate, really quickly, this pyruvate is 3 carbons, and it diffuses back through the cell walls, and it gets into the mesophyll cell again, and with a little bit of ATP added and some other stuff, it will turn back into PEP. So pretty much it recycles. Now, this pretty much means that we're going to have only CO2 down in here in the bundle sheath cell and very little oxygen down here. Now, this is really good, but why wouldn't all plants do this? Well, it's because of this extra ATP, really. This takes a lot more effort and energy, and the C4 plants are willing to take or to use the extra energy because it means in the long run they won't have to use as much again, or at least lose as much from the photorespiration. The C3 plants can't really benefit from it. So, pretty much, as a quick review, C3 plants live in cooler environments. They're kind of considered the normal plants. The C4 plants live in hotter environments, and so because they need to keep their stomata closed, there's more oxygen inside, and so C4 plants have adapted to deal with this, and effectively what it is a filter. So this PEP is extremely selective and will only bond to the CO2, which then allows this malate to be created to go through the cell wall and to combine with the less selective RUBP. Whereas up here, again, the RUBP is not selective, so if any oxygen is in here, especially if there's more oxygen, it'll just combine with it. The PEP, not so much. So, I hope this was interesting. If you have any questions, I guess you can ask me. Here are 2 examples. I thought they'd be fun. They took a really long time to draw, so I thought I appreciate them for a moment here. So corn is a good example of a C4 plant, as is a cactus. Really anything that lives in a hot environment is probably a C4 plant. So, yeah, hope you enjoyed.